Okay. So the next part will be DNA replication. And we're going to go over the whole process of actually replicating DNA. So now we're going into the cell, into the bacteria. This bacterial cell has accumulated enough energy that it's reached the point where it decides it wants to replicate its DNA and create a new daughter cell, so one cell becoming two cells. And so DNA replication is important to remember. It starts at a single point in the DNA chromosome, and this is referred to as the origin of replication. And it's also often just let's try this. And then it's often, let's get that pen back, called the oriC. So at the site of the replication, the helix is unwound, creating a replication fork. Replication is bidirectional, therefore there are two replication forks, and then a replicon is a length of DNA replicated. So let's start with this. So let's have a visual representation of just what I said. So this is a bacterial chromosome, and let's consider E. coli. So E. coli is a bacteria that you hopefully know pretty well by now. And the E. coli chromosome is about 4 megabases long. In bacteria, it's a circle, and so it's a circular chromosome, and so very often we designate the origin of replication as the 0 point, and then the place where it terminates is called the tear. And so this is about 2 megabases away. So if we set the origin equal to 0, we go 10 base pairs of 100,000 base pairs, on and on until we reach the other end, and that's about 2 million base pairs away. So there's 2 million base pairs this way and 2 million base pairs this way. So this is where DNA replication occurs, and so now you can see an actual replicon that's being produced. And so that's because this is where DNA is being duplicated. So a single double strand is becoming 2 double strands. And so this is actually progressing in 2 directions, so it's progressing this way and this way. And eventually, these 2 forks meet up, and once it hits the terminus, then we've completely replicated the chromosome. So this is why we also say it's bidirectional. It's bidirectional because even though it starts at the origin of replication, there are 2 polymerases, there are 2 forks, and so it's actually replicating DNA in both directions. So if it wasn't bidirectional, you would have a case where your origin and your terminus would be in the same place. And so you would start here, you would replicate, 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 and you wouldn't stop until you reach the other side. And so this is what we mean when we say it's bidirectional. So there are a lot of molecular factors that are required for DNA synthesis, and I'm going to expect you to memorize these. So you should know all of the enzymes, and you should know all of the molecules that are necessary for actually creating, replicating DNA. So these things include things like DNA polymerases, gyrases and topoisomerases, helicases, primases, ligases. These are all enzymes. These are all proteins. And then you also need specific chemical molecules like primers, DNTPs, and magnesium. And so these are not proteins. These are other chemical molecules that are necessary. So DNA synthesis in bacteria involves 5 DNA polymerases, and they are named in the order of biochemical isolation. So DNA polymerase I, 2, 3, 4, and 5. And so these are all enzymes that were purified out of bacteria, and in vitro were shown to be able to replicate DNA. So the first one was isolated from E. coli, DNA polymerase I, and so if they added a DNA, experimenters found if they added a DNA template, if they added all 4 DNTPs, a starting primer, so a primer is a short piece of DNA or RNA that is complementary to it, then they could get DNA replication to occur. And so the enzyme, this polymerase, was able to actually add DNTPs to it in order to form complementary sequence to the DNA template. And so this is just a visual depiction of it. So you start with this DNA template. So the DNA template is in red, and then you have a primer, which is in blue. So this is a piece of DNA that's already there, that's already complementary. And so by adding DNA polymerase I and then magnesium, so magnesium is a cofactor ion that's necessary for this ion to function, they were able to slowly extend the length of this one by one. And then they also found that they produced inorganic pyrophosphate from this, which we know because the DTP, these are used, these are necessary, but these have 3 phosphorus atoms within it, and then once it gets incorporated into the DNA, you cleave off 2 of those, and so this is the remaining chemicals, that the phosphorus pyrophosphates that are cleaved off of the DNTPs. So this study showed that the elongation occurs in the 5' to 3' direction, adding 1 nucleotide at a time. So the nucleotide was added, the 2 terminal phosphates were cleaved off, providing newly exposed 3' OH, and then this 3' OH can participate in addition of another nucleotide as DNA synthesis proceeds. And so what do we mean by this? So we have a chain of single-stranded DNA. So right now there's only 2 nucleotides, A and T. And so the first thing you want to do is incorporate this new C within it. And so it's important to note that this grows in the 5' to 3' end. So this is the directionality of how this grows. So it starts from the 5', which is the 5' carbon here, and it grows towards the 3' end, which is the 3' carbon here. And so this nucleotide is able to be incorporated, and then in the process of the bond being formed, these are cleaved off, and then this phosphate goes right here. And so now we have a strand that is 3 nucleotides long, that is A, T, C, and we have the addition of these the 2 pyrophosphates. So each of these polymerases were able to have in vitro activity, but they actually do very different things in the cell. 1, 2, and 3 all can elongate existing DNA strands, but they cannot initiate DNA synthesis. Exonuclease activity, so they all have exonuclease activity, so in the 3' to 5' end. And so they are able to actually cleave off nucleotides as necessary. So if they were actually able to polymerize this, they could then cleave off this nucleotide if they so desired. And so that's what we mean by 3' to 5' nuclease activity. We can degrade the nucleotide going in that direction. And so why do they do this? They do this as part of proofreading, which we'll get into in a little bit. And then one of these polymerase, DNA polymerase I, have 5' to 3' activity, exonuclease activity as well. And so that means it's actually able to start from this direction and then cleave off nucleotides as it so desires going this direction. And so this is probably very confusing at this point, but it'll hopefully all come together as we go over the whole process of DNA replication. So if we look at these 3 polymerases in the cell, so this kind of just summarizes what they can do, and this is the number of molecules we find in the cell. So there's about 400 of these. There's about 15, only 15 of these polymerases in the cell, and it's kind of unknown exactly how many polymerases these are. So these 2 polymerases are much more important for us. And then all of these are involved in so some of these polymerases are induced by DNA damage or other external forces that force you to need to do something in order to save the day. So the most important one is the DNA polymerase III. So this is a holoenzyme. This is the active form of DNA polymerase, and it contains both core enzyme complexes made up of a bunch of subunits. So multiple proteins are actually within the DNA polymerase. And they have different functions. So there's the alpha subunits that's required for the polymerization. There's the epsilon subunits that's required for the exonuclease activity. And then there's the theta that's required for the assembly. And then there's additional ones that are involved for the sliding clamp. So when we put all these things together, this is what the DNA Polymerase III Holoenzyme looks like. So there's two key cores, and these two key cores are actually both replicating strands of DNA. And then we have a sliding DNA clamp that's necessary for locking onto the DNA. So if we go all the way back to our picture of how DNA replication works, what we see is that we initially have a single double strand of DNA, and then this separates into single strands where new replication can occur. And so this is essentially what's going on for this DNA polymerase. So this is why it looks like it does. So you have two strands of DNA coming in, and then one of these strands is sent to this core, and then one of these strands is sent to this core. And so now we are able to actually synthesize new DNA from that. Okay. And so now we go from one strand, one double strand, to two double strands of DNA. And so that's what this alpha subunits do. So these are necessary for the 5', 3' polymerization. The tau, for example, this just is involved in creating kind of the arms between them. And then the clamps and the clamp loaders, so these are the beta, for example, and then these are part of the gamma complex. So this is what allows this is what locks on to that DNA, and then it separates that into two single strands, or it helps separate that into two single strands. So some of the key issues that need to occur during DNA replication, so you have to unwind the helix, you have to reduce the increased coiling generated during the unwinding, you have to have a synthesis of a primer for initiation, you have discontinuous synthesis of a second strand, you have the removal of the RNA primers, joining of the gap-filling DNA to adjacent strand, and then proofreading. We're going to go over all of the enzymes that are involved in this. And so the first one is the DNA A. So this DNA A factor is what initially binds to the origin, causing conformational changes, and it causes the helix to destabilize and open up. So this is the first time where double-stranded DNA, you have double-stranded DNA, and that can be separated into single-stranded DNA. And so, you still have parts that are double-stranded here and here, and then, but now you have exposed DNA, and DNA A is responsible for that initial exposure to separate the double-stranded parts together. Once you've initiated that, if you want to continue to push to expose more and more of this single strand, this is where DNA helicase comes into play. So it's made of a hexamer of this gene called DNA B, and then it just further unwinds the DNA. So it pushes this out, it pushes the exposed region out, kind of separating out the double strand pieces of DNA. And so, if we look at it, after this has happened, then we have, more exposed. So initially DNA A does that, and then this gets extended by the DNA helicase. And then to further stabilize this is where the single-stranded binding proteins come in. And so these proteins right here just bind to single-stranded DNA, and then that helps prevent the DNA to reanneal. So the reason that these strands don't reanneal with each other is that there's single-stranded binding proteins that come in and bind to them. So we've started, so we will get to supercoiling in a second. We've started by unwinding the DNA. We've separated the two strands, and now we're ready to start initiating DNA replication. So how do we do that? So when we talked about the enzymatic activity in, so in this part right here, we talked about the fact that we could take DNA polymerase, and in a test tube, we could get it to extend and polymerize DNA. But it required certain things. It required a template, and so we've now unwind the DNA and we've provided that template. But the other thing it needs is a primer. And so it's unable just to start from this piece of DNA. It's not able to just bind to this piece and then start to creating a new strand. So it can only create a strand if it already has something to start from, and that's called a primer. So it's able to extend from here. So one of the things we need in order to start DNA replication is that we need primers. And so primase is the factor that actually is able to do that. And so this is actually an RNA polymerase, and it is recruited to the replication formed by helicase, and then once it gets there, it synthesizes an RNA primer. So it's able to take that single strand of DNA, and it is actually able to synthesize a primer that's complementary to it. And I'm not exactly sure how large it is, maybe 10 to 20 base pairs long, something like that. And so now, once this primer is there, then the DNA polymerase can come in and extend from that. So starting from that RNA primer, you are able to extend from that. And so, if we try to draw this out, let's see how well I can actually draw something like this. Actually, I guess it's already right here. So we have the DNA template right here. And so the primase has come in and created a piece of RNA that's complementary to it. And now DNA polymerase III is able to come in and then extend from that. So you have the single-stranded DNA, and then you have the primer. The primase comes in and creates this RNA primer. And so now DNA polymerase III is able to extend from that. And so note the way it works is that it always goes in the 5' to 3' direction. So it starts at the 3' end of the primer, and then it creates new nucleotides in the 5' towards the 3' end. So eventually, if you look at the strand, it's eventually going to be able to go all the way from the origin to the terminus. And so an issue that now has to be overcome is that this RNA primer has, we don't want to end up with something that's a hybrid, a chimera of RNA and DNA. And so the next step that needs to happen that we're going to cover is we need to actually remove this RNA. And so what we want to do is remove the RNA and add it and turn it into DNA. And so this is what DNA polymerase I does. Its job is not to be the main source of replication. Its job is just to remove the RNA primer and replace it with DNA. So once it has this to start from, it's able to both degrade this and remove it, and so in the end, you're just going to be left with DNA. All right, so far so good, but now we need to think a little bit more deeply about the fact that DNA replication always goes in the 5' to 3' direction. And so this actually creates a problem for the cells and leads to needing to deal with the two strands separately. And so we need to deal with the leading strand which has continuous DNA synthesis, and then the lagging strand, which has discontinuous DNA synthesis. So why is this true? Why is this an issue? So here we've unwound DNA, and we've separated it into the 5' into the let's say the Watson strand and the Crick strand, and so these are binding anti-parallel. So so we have the 5' to 3' this way, and then for this one it's 5' to 3' this way. So if we synthesize DNA from these two strands, we're going to actually be synthesizing them in different directions. And so for this one, the new DNA goes this way. So the template strand, it goes from the 3' to the 5', but for the new DNA, it's being generated from the 5' to the 3'. And then if we look at the second strand, it's actually going in the opposite direction, and that's due to the anti-parallel nature. And so if you have polymerases that are locked together that are doing this, they're basically getting pulled apart. So if this polymerase would follow along on this DNA strand, eventually it would want to go in completely opposite directions. And that's not what happens. You know, that could have been what happened. It could have been life figured out a way to do it that way. But instead, life went through this completely more complicated process. And so what it does is it does one strand in the normal way. So you start from an RNA primer, and then you polymerize going this way. And this strand is going to continue on until it reaches the terminus. So you're going to actually, you know, as long as there's not some sort of error, some sort of problem, you're actually going to amplify 2 million base pairs of DNA going this way. So you'll have this small RNA primer, and then, 1.9 million pieces of DNA after that. And then again, eventually DNA polymerase I will replace the primer with the DNA. But for this other strand, the lagging strand, you know, because these things are locked together, it keeps creating pieces of DNA kind of over and over again. And these are on the order of 1,000 base pairs. So you have one strand that's just continuously synthesizing, and then another strand where you keep on synthesizing it in pieces over and over and over again. And so that way, both polymerases that are replicating can move in the same direction. So this amplifies, and then now we skip back here. Amplifies, skips back here. Amplifies, skips back here. So this one keeps moving in this direction over and over. And so it's this discontinuous synthesis that creates these pieces that, you know, require this kind of constant battle. So this RNA, the primase, has to come and synthesize this. DNA polymerase III amplifies to here until it eventually stops. Now we have a primerase come in here, it synthesizes a primer here. The DNA polymerase synthesizes until it reaches the end, and so on and so forth. And so these fragments, you know, the fact that this lagging strand has all of these fragments, these are called Okazaki fragments. So these are lagging strands synthesized as Okazaki fragments, each with an RNA primer. So each of the Okazaki fragments start at the 5' end with an RNA primer. And then as replication proceeds, you know, these slowly get replaced and removed. So the DNA polymerase comes in, and now it removes this primer and replaces it with DNA. These are still separated strands until DNA ligase comes in and basically seals these together. So it seals the DNA strands together once the DNA polymerase I comes in and replaces the primer. And so as a result, both of the DNA strands are synthesized concurrently at the same physical location. And so just to make it a little bit clearer what exactly is going on. So you have two different replication forks, and there are 2 polymerases at each point. So there's a total of 4 polymerases. So you have a leading strand that's going this way, and then you have a lagging strand that's going in pieces over here, and then you have a leading strand that's going this way, and then you have a lagging strand that's kind of going over here. And then eventually these two different things will meet once they come to the terminus. So what allows this to happen is that there's actually a looping of the single strand DNA. And so we have the leading strand, so you should be able to recognize the leading strand now because you have the primer, and this is just going to continue to go in the 5' to 3' direction. So you'll notice if I ask you to tell me which one the leading strand is, the template strand goes 3' to 5', but the synthesized strand goes opposite 5' to 3'. So this would be the leading strand. And then the lagging strand basically has to loop around, and so this allows the physical structure to remain joined, because you can't move. These two parts of the DNA polymerase are physically connected together, and so you can't actually move them around as DNA replication occurs. And so what happens is that you actually loop the DNA around on the lagging strand to allow it to synthesize in the opposite direction. Gyrase. So DNA gyrase. So this is an enzyme that reveals coiled tension from unwinding of the helix, and it's a member of a larger enzyme group called DNA topoisomerases. And so as you unwind the double strand together, it actually creates a lot of twisting. Probably past your time if you used to have a telephone with one of those telephone cords attached to it, and you would talk on the phone, and then eventually you'd hang up the phone and everything would twist together. So it's almost like that, where it just gets over twisted. And so the process of unwinding it over twists it, and then the DNA gyrase comes in and nicks it, and then allows it to rotate to basically unwind that supercoiling. So the final aspect of DNA replication is proofreading and error correction, and so this is an integral part of DNA replication. So DNA polymerase is not perfect. It's trying to do this very fast. It's adding about 1,000 new nucleotides every second to the newly synthesized strand. So think about how fast that is, 1,000 base pairs per second. And so it doesn't always get it right. It tries to incorporate the right nucleotide, but occasionally it incorporates an incorrect non-complementary base pair. And so, you might have, a CGC, and what's supposed to be there is a GCG, but instead maybe you did a GCA or something like this. And so you have this non-complementary set of nucleotides. And so these can be recognized. And so these are recognized by, so this should say 3, 1, 2, and 3. So all of these are used at different levels in order to recognize when something goes wrong. So DNA polymerase III, as it's adding, it might realize it just covalently bonded something incorrect, and so it's able to actually cleave off that A. And similarly, the 1 and 2 polymerases are involved as well. And then 4 and 5 are kind of last-ditch repair machines if things really get messed up, so they're normally not involved in DNA replication and repair. But if something goes wrong during the process, like if all of a sudden they're hit with a bunch of DNA damaging agents like UV light, where you're breaking double-stranded DNA all over the place, these polymerases come in and try to force replication to complete so that the cell doesn't die. So all this together kind of leads to a coherent model of DNA replication. So you have helicases that help unwind this DNA together. you have single-stranded binding proteins that stabilize and prevent the single strands from coming back together. These strands get fed into the two DNA polymerases that do replication. One of these is the leading strand. One of these is the lagging strand. The lagging strand is doing this in pieces, which eventually all gets ligated together. So all of these pieces eventually all get ligated together into a supermolecule. DNA gyrase comes in, and they're there in order to release the unwinding, release the winding, and the supercoiling created by the helicase unwinding. So by unwinding these two things, it's creating supercoiling downstream. So I think a lot of this is easier to see visually.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus.
We're going to skip to the point.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand.
So you can see the old strand is coming in from the left.
The DNA to be copied enters the production line from bottom left.
And one of the strands basically feeds directly into one of the polymerase, and DNA replication
occurs. As fast as a
occurs.
jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time.
So you can see that there's these sections that keep getting produced for the lagging strand. And you can see primase continue to come in. The primase is creating a new primer for DNA polymerase to act on, and then the polymerase acts for a certain period of time until it reaches the end of the previous strand. And then that process kind of goes on over and over and over again. And so,
and can be seen spooling off
as
to
a
the
result,
right.
you have this exchange over and over again. So there's
Things
one
are not
simple
so
strand,
for the other strand,
the leading
because it
strand,
must be copied
complicated
backwards.
strand, the lagging strand.
It is drawn out repeatedly in loops and copied one section at a time.
Alright, so that is a model of DNA replication in bacteria, kind of going through the whole process. So we are going to switch gears next to look at eukaryotic DNA replication and just talk about some of the key differences there. But this is also kind of exciting from a biotechnological perspective, because now we've encountered one of the first and one of the most important members of the molecular toolkit that we use all the time in lab, and is used all the time in order to generate new medical techniques, medically related techniques, and that's the DNA polymerase. So the DNA polymerase allows us to copy DNA, and we have hijacked that in order to do things that we're going to get more fully into in Chapter 20, but we're also going to start to touch upon that in the activity as well.